So the surprising fact also was that because of this clamp here, you have an increased exonuclease resistance of the small interfering RNA. One big problem when using the small interfering RNA as a drug, injecting it in the serum, is that in the serum you have a very high amount of RNases. Because actually RNA is actually not tolerated in the organism. Our chromosomes are DNA. And RNA just exists like roses at the time of a message. Just have a message, it's a messenger RNA, and then in the cytoplasm it will be degraded totally. And the only organisms, well, they're not organisms, the only chemical, let's say, yes, organisms, like maybe uses that have RNA in their genome are viruses. So in our serum, we have a very high amount of RNases, and that's why when you inject naked siRNA in the serum, they will be directly degraded. You can see it here is an experiment that we did with a Tuschl design, so it's with a classical overhang in comparison to the riboxx, incubated them in mouse serum 80%, 10 micromolar, a quite high concentration of small interfering RNA, and looked in a native page what happens with the siRNA incubated after 1 hour, 2 hours, 4 hours. And you can see that with the Tuschl design, you have a complete degradation of the siRNA after 6 hours. In comparison, the riboxx siRNA is resistant up to 48, I will say up to 72 hours, because of this clamp that increases the resistance to exonucleases present in the serum. So this was also very important for us when we went on doing in vivo models of infection. The next question was, and all chemists will tell you, this, to synthesize this in a chemical synthesis, it's a pain in the neck, because those 4 Gs will form tetramers if you are synthesizing it on fast phase, on solid phase chemistry, excuse me, and those tetramers will lead to, in the analytic phase or in the purification phase of your product, chemical product, to a product that you won't be able to analyze or to prepare. So we looked at a way to synthesize this double-strand RNAs with those Gs, and we used an enzyme, the calicivirus polymerase, and this actually is a very simple device. You just have, you synthesize the template with a 5C, where you have your seed sequence. Like a PCR, you add the polymerase, ribonucleotide, buffer, and it's not a thermocycling, it's 30 degrees for 2 hours. So it's an isothermal method. And you get your 2 strands perfectly annealed with your Gs at the end. And this is a little bit how it goes. It's really straightforward. You can also do it in 1 hour. It's also working very fast. And this is how it looks like. So you have this template that you have to synthesize in a chemical way, analyzed by anion exchange, the products after purification. And this is how it looks like on a polyacrylamide gel. So you have here your template. This is a marker of 17 base pair, 21 or 25 RNA base pairs. And the product, the double-strand RNA product that you get with a perfect annealed product that is, after 1 hour, ready to be used as a small interfering RNA. So really a straightforward method to synthesize RNA. Now, looking at the inhibition of the rhinovirus in vitro, the method was, the experimental setting was the following. We used HeLa cells. Those were infected with the human rhinovirus, 1b, 10 to the 4th TCID50, so there are about 10,000 viral colonies, if you wish. And in parallel, synthesized the siRNA by the biocatalysis method. That's with the polymerase, purified it, analyzed it again, and put it on the cells, transfected it with liposome-based solutions, and measured the EC50 and the CC50. The EC50 is, as you know, the concentration that protects the cells in 50% of the wells, because you're infecting with the virus and you're protecting the cells by giving siRNA directed against the viral genome. But of course, you have to look at the cytotoxicity of this RNA, because it's nucleic acids who could also have, like any drug, a toxicity, and measured both of them. So this is here are the results of viral genome, and the siRNA is directed against different portions of the genome. In blue, you have an siRNA sequence according to the Tuschl rules with the overhangs, and in orange, you have the same sequence, everything the same, except that you have a clamp as a GC at the end, and it's blunt. So the first message is that if you have a very strong secondary structure, like here at the beginning of the viral genome, it won't work if you use a Tuschl design or if you use a riboxx design, because you're not getting inside of this very strong third conformation that is in iris. But you have some cases like here that if you are using a tutorial design with an EC50 of 500, just take the same sequence, add the riboxx motif at the end of your siRNA oppositely to the seed sequence, and you get your EC50 of about 75. Here it's the same. Here it's the same with this 9 siRNA. You are about 300 approximately. Here you are really getting really down. You need much less siRNA to have the same effect. This was the control luciferase and so on. This is a Western blot showing the same results again. It's a little bit similar to what Dr. Mziu showed with the ICA 512 with two different manufacturers and the riboxx design. We looked here at the expression of the viral protein 1 in the cells using the different siRNAs according to the tutorial design with the overhangs with 20 nanomolars. You can see we cannot knock down the viral protein. Using the same concentration, the same sequence, but adding our motif, you have in almost all whales a completely knockdown of the VP1, except here there's some protein left. But you can really go a little bit, like 50 nanomolars, you will get complete rid of the protein. So you have really an increased potency and efficiency of this RNA. This is really important if you are going to perform RNAi screens. So screens with this RNA, you need to take care of target effects to increase the specificity of your effect, and very important, of course, if you are going in animal models. Those are the controls of beta-actin. So having selected 4 siRNA that are active in vitro in our bottles, in our lab, we went on and asked the question, is it possible to see the same effect with the riboxx siRNA in mice? And the experimental setup was the following. You take the virus, purify it, have it 10 to the 4th or 1 million TCD50, and you inject this amount in 20 to 30 microliter intranasally. You instill it intranasally in the nose of the mouse, and the mouse will inhale it and bring the virus, as shown in the first slide, directly to the lung. And we looked at, after infection of the mice, so they are very young mice, 18 grams, we looked at the viral load in the lung in the lower left lobe. So it's really gone. You're not looking here in the oropharynx, you're looking really here if you are getting some virus. And we did it with qPCR normalized against beta-actin. This is a time scale, 0 to 168 hours, and you have the viral load here. Using the 10 to the 4th doses, you have an increase of the viral load, and then after 2 days, disappearance, clearance of the virus. If you go with the higher doses, you will have a plateau until 120 hours, and then after 7 days, you have a clearance of the virus. So this model showed us, okay, we can go on and use now this model to test the effectivity of the riboxx siRNA that were working in vitro in the cells. And that's exactly what we did. So synthesized the siRNA by biocatalysis, purified it, analyzed it, and then injected 1 milligram per kilogram. So it's 1 microgram per gram. We are talking about 18 gram mice. So it's like 20 microgram, 2.5 nanomole of siRNA intranasally with a liposome-based solution, and looked after infection of 1 million TCD50 intranasally also in parallel, looked at the viral load against in the lung. And those are the results. So you have here in black the viral load without treatment of the mouse. In green, you have the viral load by treating the mouse with an siRNA that is not directed against the viral genome. So you don't see an effect in comparison to the viral load where you do not use any siRNA. I must say that those are 4 different measurements. So you have, for each point, you have 4 different mice that were used for measuring the viral load. And in red, you have the treatment of the mice with one of the siRNAs, the riboxx 10. And you can see that already after 72 hours, you have a decrease of the viral load in comparison to the controls. And then at 96 hours, you have a complete clearance, disappearance of the virus from the left lobe, lower left lobe of the mice, indicating that at this time point, the siRNA is also active in this mice model. So as a conclusion, I hope that I'm on time. So it's possible to target the human rhinovirus, its genome, and to inhibit its replication in vitro and in vivo. And thanks to a new design, a really innovation, I must say, we have patented this in the synthesis using biocatalysis, allowing to get this new riboxx design with this GC clamp at the end that is highly efficient and is resistant to serum RNAs and endonucleases. We could also have an inhibition of the rhinovirus infection in mice model with 1 milligram per kilogram. And what we're working on now is to allow large scale, or our objective is to go through large scale synthesis now of this siRNA. For example, if you need 1 gram or up to 10 gram, to use this biocatalysis and to allow this production in a cost-effective manner and a quite easy manner of those small interfering RNA for therapeutic purpose. So those are the guys now working at riboxx. We are located at Radebeul in the Pharma Park, where actually AVD at the beginning and then Albion, and now they're named Biotai, so there have been many fundraising turns, and the names were changing each time. So, but we are also called, we are still called riboxx. So I hope that this will remain the same. There's a guy working in the lab. The animal models were mainly performed by Julia Gebhardt, who is working mainly in this field of animal models with the rhinovirus infection. With Romy Zieger, who was not there at the beginning. She was looking for after her child. She went pregnant in the middle of our experimental setup, and so she couldn't be there on the photo. But Julia and Katrin did the animal experiments, and those are the guys working in the production. Dr. Zimmermann, Christiane Petzold, Constanze Grüner and Christine are doing the synthesis, are doing the chemical synthesis. So we have a very nice synthesis facility now there, and the other people are administrative, and thanks you for your attention. Be happy to take some questions. Yeah, please.
I would be interested if these siRNAs, double-stranded molecules, do they induce interferon alpha or beta?
So we have tested this for this because, of course, it's a polymerase-based synthesis, and you have a triphosphate as a 5' of the genome. And this is a work that was done by Professor Schmidt, by Mark, there, and Rebecca. It was actually a burning questions. We're asking ourselves, what do we see a non-specific clearing of the virus, or is it specific? And up to, I think, 250 microgram using DCs, I think was SLAN DCs, we didn't see any activity with using those 4 siRNAs. So fortunately, it's yes. Well, we are now, so as I said, we use 20 micrograms, so up to 250 in vitro, we didn't see anything. Yeah, please.
Maybe I missed it. How have you injected the siRNA?
Intranasally.
For the distribution of the siRNA molecules in the whole mouse?
No, we didn't. So it was a local application. We've chosen also the rhinovirus model, not only because I'm a virologist and an RNA virologist, but because, of course, we didn't need this delivery to address this delivery aspect, the systemic delivery aspect, but went for, let's say, an easier, so low-hanging fruits, and said, okay, let's just do local administration of the siRNA with lipofectamine. So it wasn't systemic. This is really a big aspect that has to be dealt with.